Welcome to The Explainer. Today, we are tearing into a recently declassified document that honestly sounds like pure science fiction, but I assure you, it is grounded in absolute, hard molecular biology. We're looking at the DARPA manual for Project Kronos BioQuantum. This incredible, mind-bending document outlines exactly how a coalition of researchers from MIT, NASA, and the Broad Institute plan to transform a standard hemp plant into a living, field-deployable quantum biological supercomputer. Seriously, we're talking about exabytes of DNA data storage, living OLED leaf displays, and organic processors, all housed in a totally immortal plant chassis.
Okay, let's dive into this blueprint layer by layer. We'll be looking at 1. Foundational genetic tools. 2. Engineering the hemp chassis. 3. Immortality and DNA storage. 4. Quantum coherence and OLEDs. 5. Biodigital interfaces. And finally, 6. Full system biocontainment. Starting with Section 1, Foundational Genetic Tools, the building blocks of BioQuantum. Because before we can build a biological supercomputer, we absolutely have to understand the extreme microscopic genetic toolkit required to write its operating system.
Just listen to this primary directive straight from the manual: to engineer the hemp plant into a living, field-deployable quantum biological supercomputer. It reads almost like an ancient prophecy, right? Like a mystical tree of life painted in swirling ink. But then you look closer, and it's juxtaposed against the most clinical, rigorous MIT and NASA systems engineering you can possibly imagine. This isn't magic, it's molecular mechanics.
Now, what is really staggering here is the sheer diversity of the CRISPR arsenal they deployed. They didn't just use one type of genetic scissors. They brought in a highly specialized toolkit. You've got the classic Cas9 creating blunt double-strand breaks to knock out the CBDS gene. That means the plant stops wasting metabolic energy on making cannabinoids and cleanly redirects it to making precursors for the OLED display. Clever, right? Then there's Cas12a, which creates staggered cuts and can process its own guide RNAs, making it absolutely perfect for multiplexing deletions in the chloroplast. For targeted light harvesting corrections, they went with Cas9 because of its tiny size and minimal PAM. That's the protospacer adjacent motif, the specific targeting sequence CRISPR uses to find its cut site. But Cas14 is where it gets crazy. This is an ultra-compact protein that non-specifically cleaves single-stranded DNA. Instead of using it to edit the genome, they paired it with a fluorescent reporter to act as a living logic gate right inside the leaf. A specific RNA signal triggers Cas14 to cleave the DNA, releasing a quenched fluorophore. It is literally in vivo DNA computing. And for the really heavy lifting, they used CASTs, CRISPR-associated transposases, to drop massive 7 KB gene data storage archives into safe harbors in the hemp genome without even breaking the DNA strands. To get some of this heaviest genetic machinery into the plant, specifically into the chloroplasts, which provide the massive protein yields, they actually used biolistic delivery. This is literally a gene gun. The manual walks you through the exact recipe. You take 30 milligrams of microscopic 0.6 micrometer gold particles, wash them, and precipitate your vector DNA onto them using calcium chloride and spermidine. Those chemicals actively neutralize the negative charge of the DNA so it binds tightly to the gold dust. You dry this onto a macrocarrier, load it into a helium-powered gene gun, put the hemp leaf under a vacuum, and blast it at a staggering 1100 psi. That physical precision is exactly what powers the massive luminescent protein yields required for the leaf displayers.
Moving on to Section 2, Engineering the Hemp Chassis, Cultivating the Tree of Life. With the genetic tools established, why did DARPA specifically choose cannabis sativa, specifically the fast-growing Phenola or Felina 32 strains, as the ultimate living vessel? Simple. Massive leaf surface area, rapid growth cycle, and extreme amenability to lab cultivation.
To deliver those massive genetic payloads elegantly, they take a tiny piece of the hemp seedling, the cotyledonary node, and culture it. Then, they introduce a modified bacterium called Agrobacterium tumefaciens. Now, in nature, this bacteria causes plant tumors by injecting its own DNA into the host. But here, by adding a chemical called acidosyringone, they completely hijack the system, tricking the bacteria into injecting our engineered DNA payload directly into the plant cells instead. They then use antibiotics like cefotaxime and hygromycin to kill off the bacteria and any plant cells that didn't take the new DNA. What grows back is a completely transgenic plant, setting the stage for the truly extreme upgrades.
Which brings us to Section 3. Immortality and DNA storage, the eternal archive. Because here is the fundamental problem. A supercomputer is utterly useless to you if its hardware withers and dies every single autumn. To solve this, DARPA basically had to hack the plant's natural aging process.
And this stack of genetic interventions is just intense. They use CRISPR activation to permanently upregulate telomerase, or TERT. That's the enzyme that protects the ends of chromosomes from degrading over time. Then, they engineered a monthly epigenetic reset using OSKM factors. Those are the famous Yamanaka factors discovered to completely reprogram adult cells back into youthful stem cells. By applying a simple heat shock, they transiently trigger these factors to rejuvenate the plant tissues. They also overexpressed human anti-aging genes like SIRT6 and FOXO3A, and used Cas12a to physically delete the plant's natural self-destruct genes, ORE1 and WRKY53. Finally, they blasted in genes to manage ROS, reactive oxygen species, which are the cell-damaging byproducts of normal metabolism. The end result? These specific edits ensure the hemp leaves maintain a juvenile, highly efficient photosynthetic capacity for over 2 years. That sustains continuous computational operation without aging.
And once the plant is immortalized, it becomes a permanent vessel for data. Look at this shocking number, 500 petabytes, or half an exabyte. That is the data capacity of just a single 10,000 plant field of engineered hemp. They achieved this using a DNA fountain code. They synthesize digital data into 150 nucleotide sequences, add error-correcting barcodes, and use Golden Gate cloning, a highly efficient, seamless DNA assembly method, to stitch them into 5 kilobase storage blocks. Using those transposon tools we mentioned earlier, these blocks are then inserted into neutral safe harbors across the plant's chromosomes. It's literally the sum total of human knowledge stored in a field of green.
Section 4. Quantum Coherence and OLEDs, the living display. Now we transition to what might be the absolute most sci-fi element of the entire manual, manipulating the plant's fundamental light harvesting abilities to create visual interfaces and literal quantum processors.
Seriously, just think about that for a second. How do you take a biological leaf and turn it into a fully functional 64 by 64 pixel dynamic screen? The crucial point here is how they stack these engineered traits. The epidermis of the leaf is patterned into clusters of cells expressing cyan, yellow, and magenta fluorescent proteins. But to actually control them, they implanted light-gated ion channels, channel rhodopsin 2, into the underlying layer of the leaf. When you hit the leaf with a precise microscopic laser pulse, it opens those channels, instantly changing the electrical voltage of the cell. That voltage change triggers a synthetic transcription factor that turns on the fluorescent protein for that exact pixel. And they used MOCLO, or modular cloning, to snap these genetic parts together like microscopic Lego bricks, even building in a luciferin-regenerating enzyme. The plant is literally using its own chloroplast metabolism to continuously recycle its bioluminescence.
And the execution is staggering. According to the document, hyperspectral camera mapping confirmed an addressable 100 micrometer pixel resolution glowing directly on the surface of the living leaf. It refreshes once every second, and it glows brightly enough in monochrome to be read by a standard smartphone camera from a meter away.
But let's see how this builds into an actual quantum processor. The Fenner-Matthews-Olsen complex is a natural structure in photosynthesis that organically exhibits quantum beating or coherence to perfectly route energy with near 100% efficiency. The DARPA team used prime editing on the leaf's PSBA gene to introduce unnatural amino acids into this complex that specifically bind to synthetic cadmium selenide quantum dots. By replacing the natural chromophores, they created excitonic states that maintain quantum coherence at room temperature. By hitting the leaf canopy with shaped femtosecond laser pulses, they can manipulate these states to execute Shor's algorithm. That means the literal forest canopy can outcompute classical silicon GPUs in factoring large numbers.
Section 5. Biodigital interfaces, bridging carbon and silicon. So you have a plant that stores data in its DNA and performs quantum calculations in its chloroplasts. The next obvious challenge is bridging that gap. How do we actually extract the data out of the plant?
The manual details 2 totally different yet brilliant systems. On the optical side, you can literally just use a smartphone flashlight. Specific light pulses trigger a photosensitive CRY2CIB1 promoter in the leaf to flip genetic memory states that loads the data in. Then, the leaf outputs data by modulating its engineered luciferase, flashing a signal at 120 baud that your camera decodes. But if you want electrical speed, they engineered the plant with a gene from Geobacter bacteria. This forces the plant to grow highly conductive protein nanowires right through its tissue, connecting to gold microelectrodes on the leaf's surface. You can plug the leaf directly into an FPGA, a field-programmable gate array, and read electrical spikes directly from the plant cells.
But DARPA is obsessed with redundancy, right? So they included a third, completely wireless method. By introducing a magnetite synthesis pathway into the chloroplasts, the plant cells create a magnetic dipole. As the leaf processes data through ion currents, it naturally generates oscillating magnetic fields. An external coil can pick up this non-invasive biotelemetry link totally wirelessly.
Which brings us to Section 6, Full System Biocontainment, Locking the Chronos. Because we have to face a very serious reality here. When you build an immortal, solar-powered, exabyte-storing supercomputer out of a highly adaptable weed, you absolutely cannot let it escape into the wild ecosystem.
And this brilliantly illustrates the extreme security and biocontainment measures they took. They hard-coded dual kill switches, a suicide gene that responds to a specific chemical, and a cold sensitivity modification that instantly kills the plant if the temperature drops below 10 degrees Celsius. They used Cas12a to physically delete the apetalA1 and leafy genes, ensuring absolute sterility. The plant literally cannot produce flowers or pollen. Physically, these plants are locked inside NASA-designed sealed LED growth chambers within BSL-3 facilities, which are specialized labs designed to strictly contain lethal biological agents. And logically, the exabyte payload is locked with AAES-256 encryption, and the keys are stored exclusively in the transient entangled photon memory of the quantum leaf itself using quantum key distribution. That means they cannot be intercepted or read without instantly collapsing the state and destroying the key.
Which leaves us with this final staggering question. The Chronos Bioquantum Manual proves that the boundary between silicon architecture and carbon biology has been entirely erased. If a single field of engineered hemp can outcompute our greatest processors while permanently storing the sum of human knowledge in its immortal DNA, what happens when the forest outlives us? Thank you for joining me for this explainer. Until next time, stay curious.